thank you very much for that introduction, and thank you to the organizers, all organizers on different levels that have set up this meeting and also invited me to come here and speak. So my name is Pernilla and I'm from Sweden, and I will also talk about proteins. I will give you my view about proteins and connect and complement the two talks that you heard in the morning. But first I want to see in here, is so many young people. How many of you are high school students? Raise your hand. Because we really do this for you. And my talk here is really for you high school students to inspire you and tell you about what we think are exciting in research. And this is my first time in Hong Kong, I also must say. So this is very exciting to be here for many different reasons. So I will talk about proteins, and I call it protein folding as a basis of life and death. And I will tell you what I mean with that. So I will take you down in scale now, so we come to the proteins. If you think about living organisms, human beings, animals, that we can see and touch, they are built up of cells and hold together by many, many cells. There are millions and millions of cells in a human body. In these cells and outside the cells are the proteins. They are tiny, tiny, but we have a lot of it. In a human being, about 20% of the weight is proteins. Think about how many kilos that is. So these proteins, they're tiny, but we have a lot of them. They are the molecular machines of life, as I call it. They do all the work to keep us alive and to make us function in all different ways. And this is where my research centers. And you heard about key proteins already. Antibodies are proteins, for example. And Alan first talked about many different proteins too, and you'll hear some names come up here again. But before I tell you about proteins and what they do and why I think they're so exciting, I want to tell you about my background a little bit. How I became this professor I am today. And to do that, I show you a map, so you can see where I've been. So I'm Swedish. I was born in Sweden, and I moved around in Sweden, and I got my PhD at this university called Chalmers University in Gothenburg. And I studied physical chemistry. Then I moved to the US, and I did a postdoc at Caltech in California. And then I stayed in the US, and I became an assistant professor and then associate professor at a university called Tulane University in New Orleans, in Louisiana. And then I was recruited to another university in the US, Rice University, that's in Texas, Houston. And then after some years there, I had two little kids, and we decided that actually we wanted to move back to Sweden, truly to raise the kids in Sweden, where we came from. So I went to the north of Sweden, Umeå University, and I was a professor there in chemistry. Until now, since many years back, since 2015, I was recruited back to Chalmers and became a professor there since 2015 in a department that's called Life Sciences. So that's kind of my path. Oh yes, and I had a picture. My kids are now grown up. They are your age, high school students. And they are then in Sweden. So we will talk about proteins, and as I implied in the title, most proteins must fold in order to function. So where do proteins come from? The proteins that we have in our bodies. Well, protein comes from food. We eat food, and most food, cookies and everything you had out there now, they contain some amount of protein. That protein is broken down to amino acids, or the building blocks in our bodies, in the gut. And then those building blocks are taken up in the cells in our bodies. And all these different millions of cells, the building blocks go out there and then go into these cells. And in all these cells, new proteins are made, the proteins that the body wants. And then we have 10,000 different proteins in every cell or more that perform all mechanical, chemical, physical work that are needed to kind of keep organisms alive. So what proteins are made in each cell is dictated by the genetic information, the DNA. That has the information that's transcribed into RNA that's translated into a polypeptide chain, or a chain of amino acids, kind of like my rubric snake that I have here. So you put amino acids in a linear chain, and that's the peptide chain. This chain cannot function like this. It has to fold up into a unique compact structure in order to get a shape or a folded structure that dictates its function. This is what we call protein folding, when you go from this, linear chain, long, that folds up into something unique. And we have many different folded structure, unique structure. It depends on the linear chain of amino acids, what order you have put these amino acids in. And the different shapes give the different proteins the different functions. And this type of folding is what I focus a lot of my research on. And folding can go wrong sometimes. If we don't have correct folding, we can have misfolding, and that can lead to aggregation and degradation. And problems with folding can be due to mutations in the DNA that puts the wrong amino acid in a particular place in this chain, and therefore it cannot fold correctly. Or it could just be perturbations due to stress and aging that mess up the protein. So when the unfolded protein is made on the ribosome, when it's in a cell, it's released as this long floppy chain, then the protein, as Alan first told us, spontaneously fold most of the time to this folded state. And I sometimes mimic this as going from kind of a boiled spaghetti, or a boiled noodle, maybe it's better to say here, but just think about something floppy that then roll up to like a yarn ball that is the folded state. But we can also use these rubric snakes to illustrate this. Proteins fold spontaneously because the folded state is the most energetically stable state at physiological conditions. So somehow there's a driving force, an energetic driving force, to go to the folded state. And we know for a long time that all the information for how to fold is somehow encoded in this linear chain of amino acid. The order of amino acids here dictates what the final folded structure is. But how does this work? Folding is fast. It happens in us all the time now. But if it were a random process, it would have taken forever, because there's so many degrees of freedom, this Leventhal paradox that Alan first also mentioned. So there must be something that guides protein folding. There must be pathways or guiding factors. And to study that, we can do time-resolved experiments, kinetic experiments, to look at the reaction progress. And I'll tell you more about that. But I want to point out that we have made important progress, amazing progress. And this year's Nobel Prize that Alan also mentioned, the Chemistry Nobel Prize 2024, went to computational protein structure prediction and design. And really what these people figured out is that they could use artificial intelligence to be able to just look at the amino acid sequence of a chain and say what final structure that will become into. And that's really amazing. Now we know how all proteins in the world might look like. But it doesn't say anything about the pathway, about the folding mechanism. And that is also important to focus on, because most of the time protein folding works well, but sometimes it goes wrong, and that can lead to diseases. Protein misfolding or protein not folding can cause human disease. So it can lead to loss of function type of diseases. If the folded protein is not made properly, then you lose that function, right? And that can be an important function. That is many times the case in cancer, and tumor suppressor proteins like p53, if they have mutations and they can't fold, we don't have a tumor suppressor function, and therefore tumors can grow. But we can also have amyloid type of diseases or gain of function diseases where proteins aggregate into ordered structures or ordered fibers. So I have two of these here. So if you have proteins, more than one of these, and they start to aggregate with each other, you can form these long aggregates or fibers or plaque, as we many times call it, that cause disease. And if you pack these rubric snakes like that, but they have a beautiful beta strand structure. So it's not a disordered aggregate, it's a very ordered aggregate. This type of amyloid formation is the basis of many diseases we know of today. Most importantly, or most well-known, are probably Alzheimer's and Parkinson's, but also Huntington, prion diseases, ALS, and even type 2 diabetes, that is not a neurodegenerative disorder, also involves amyloid formations. We know of over 50 diseases that relate to amyloid formation. You can kind of mimic it a little bit like boiling an egg. When you boil an egg, the proteins in the egg, they are folded and in solution before you boil it. When you heat it up, the proteins unfold, and then they tend to aggregate with each other. And that results in this kind of network of aggregated proteins that are stuck there. So when you cool it down, you still have these aggregates, and the egg is hard. And that's why it's easier to eat a boiled egg. But the difference is that in these diseases I mentioned here, it's an ordered aggregate. I want to point out this is a huge problem. The largest risk factor for most of these diseases is age. And the population in the world is getting older. We live longer. And that's really good, right? But we want a healthy life. But we are foreseeing a lot of people, more and more people, getting these diseases in the future because we just have an older population. And we have no cures. That's the scary part. We only have some medicine that can help with symptoms for a while. So here we need to do a lot of research. And I think that understanding correct folding is the first step to reveal why it then goes wrong. So we need to know as much as we can about correct folding first. So how does protein folding work? Well, Professor First told us a lot about how it works and how we can study how it works. And I've also done many of those types of studies. But water, of course, is important. Everything is happening in a water-based solution, all life. So you have something called the hydrophobic effect. You know, what's hydrophobic wants to be inside and what's hydrophilic wants to be outside. But it's more to it. And during the last many decades, there's been many protein folding studies in test tubes. And we've learned a lot about different types of mechanisms and how fast proteins can fold. Here is an example of these 5 values that Professor First talked about, color-coded in different ways. And that's a great way to look at how things look like during the folding process. One of the proteins I studied actually folded really fast. So I have the world record in fastest protein folding, if that means something. But my protein was folding close to the speed limit, which is around 1 microsecond. But we also need to think about that most proteins in our bodies are more complicated than one single polypeptide chain. We also need to consider protein-protein interactions when we talk about oligomeric proteins. Dimers, for example, you have 2 polypeptide chains. Do these chains fold first and then they assemble? Or maybe they assemble first, and that assembly is what triggers folding. Also, we have metal-binding proteins, metalloproteins. Many proteins in a human body binds a metal ion, a small atom that gives the protein more chemical reactivity, I would say. Actually, almost half of all the proteins, or at least half of all the enzymes that we have in our body, bind to a metal ion in order to get more function, or its supposed function. For these proteins, do the metal bind first and that triggers folding? Or do the protein folds first and then the metal comes in after? We didn't know that. But with metals, it's important really to think about how they handle in a cell, because we need metals in our bodies, but only small amounts. And free metal ions are many times toxic because they are so reactive. So these more complicated proteins, oligomeric proteins and metal-binding proteins, became my focus when I started my own independent research career. I wanted to understand how these proteins folded. So I want to give you one example that kind of illustrates these extra factors. Hemoglobin. You heard the name already in one of the previous talks. Hemoglobin is a protein in blood, and blood is red, you know. And it's red, why? Well, because hemoglobin has iron as a metal bound to it. So it has a metal. It also has 4 polypeptides. It's a tetramer. So it's an oligomeric protein and it's a metal binding protein. So it kind of illustrates all these complications in a very common protein. That's really important, right? Hemoglobin transports oxygen and it's really essential for life. So over the years, we've studied many different protein systems, you know, proteins with different shapes, with different metals, to try to understand general features of these reactions. And we found that protein-protein binding can precede protein folding for oligomeric proteins, so that initial interaction might be important. We also found that metals can actually bind to unfolded protein, and that can accelerate and trigger folding of them. So if we want to understand protein folding in an in vivo scenario, we need to understand, to take these additional factors into account. And yes, we want to understand protein folding in vivo or protein behavior in vivo. And then I want to add one more complexity here, because in a cell, it's highly crowded. It's jammed with different proteins, membranes, DNA, and all kinds of things in a cell. It's very crowded. So you can see here an illustration of many, many proteins packed together. So it's not at all this dilute water-based solution that we like to use in our test tubes that helps our experiments, because it's easier to study things in a dilute solution. And this will affect, there will be less space to be in, but it will also affect the water properties. So there are many factors that can play into account, but at least you're jammed in there. So we started to study this too, how the crowding of a cell, we try to mimic that in our test tubes to see what effects that have on protein folding. And indeed, it does affect protein folding, both speed and even the final folded state of the protein might sometimes change in this crowded environment. So over time, this has led us to ask new questions. How are metals delivered to the right proteins in a cell without toxic side effects? You don't lose it to the environment and then it can go on and damage the cell. And what type of unwanted interactions between different proteins can happen in this really jammed situation? And that might be really important for these aggregation amyloid-related diseases, where proteins tend to come together and aggregate. So over the years, my research has gone from more trying to understand fundamental principles of protein folding to trying to address more directly what goes wrong in human diseases. So we are now working on molecular mechanisms of how copper ions are transported in the cell before they come to the target proteins that are needed for function. But there's an elaborate pathway of proteins that move around this metal ion and other metal ions inside cells. How does that work? And here we stumbled on a role in cancer. And the other direction that we're taking is that we go to misfolding and amyloid formation. We're trying to understand how these reactions happen that are much more complicated than just protein folding, because you have many pieces coming together. But how are these reactions modulated by other biomolecules that are there in the cell and can come in and perturb? And how can we kind of modulate this or stop this? And this, of course, is related then to neurodegenerative diseases. So I'm going to give you examples of each of these two directions, but first I want to tell you how we study proteins. We need to have proteins in order to analyze them, right, in the lab. So we make proteins making use of recombinant DNA technology that you heard of, but we use bacteria to make proteins for us. So the central dogma of biology, we take the genetic code for the protein of interest, we put it in a piece of DNA called a plasmid, and we insert that into bacteria. Then we grow this bacteria. And bacteria is stupid enough to make proteins out of any DNA it has in the cells. So it will make protein out of all the genes that's there, including the gene that I put in there. So it will make a lot of my protein. So we grow the bacteria, and then we separate, we splash open the bacterial cells, and we separate all the different pieces in there using different chromatographic steps. That means that we can get individual species one by one. And in this way, we can separate out our protein alone from the rest of the things in the cell. And this way, we can study almost any protein we want. We just put in the DNA for that protein in bacteria. And we can also make strategic mutations. If I'm interested in what residue binds the metal, I can engineer my protein on the DNA level and then express a protein with mutations at the different sites that I'm interested in and see how that eventual protein behaves. So this way is how we made a lot of different proteins. Then we have to do experiments. And we mostly do experiments in test tubes using different biophysical techniques in the lab. But how do we see what happens? You can't see a protein. I said they were tiny, right? And if they bind a metal, they have some color, but otherwise they have no color. So, and I have them in a water-based solution. Well, so we use methods to indirectly see what happens, and we use a lot of spectroscopic techniques, absorption, fluorescence, circular dichrys. I'm sure you heard of some of these methods. But this is a way to shine light on the sample and see what happens to that light and how the protein perturbs it in different ways. And that can indirectly tell us if the protein is folded or unfolded, for example. But we also use biochemical methods, calorimetry methods that measure heats of reactions, and microscopy more and more, because these amyloid fibers, they are so long, so you can see them under the microscope. This is the amyloid fibers in Parkinson's disease. And nowadays, we also try to go more and more to live cell studies to look where is our protein inside the cell. So over there, you see a cancer cell that we look at. So it's really like doing a puzzle. Each experimental method contributes a piece to the puzzle. And we need a lot of pieces to kind of see the image that comes up. Okay, with that, I want to tell you a little bit what type of discoveries we can do. And first, I want to tell you something about a copper-binding protein and how that links to breast cancer. And of course, we also heard already, and you know that first thing, that cancer is a very important disease that happens too many times. And we also have medicines now that help towards cancer in many ways. But still, patients die from cancer. So we need new approaches if we can find them. And I focused on breast cancer, and it's one of many cancers, right? But many women get breast cancer. It's very common. Actually, 1 in 8 of women will get breast cancer during their lifetime. So I think it's very important if we can find new approaches to target these diseases. So I said we study copper transport and copper binding proteins. And this one central protein here is called ATOX. all proteins have funny names, but ATOX transports copper in our cells and moves it around to give it to targets on enzymes that need copper. So if you can go into YouTube at a later state, you can, and I think you can do it here in Hong Kong, if you look for this movie, Copper Ion Transport in Human Cells, that's the name of it, and I'm the author or maker, you can get a short 2-minute video of the basic steps of copper transport in a human cell and what type of mechanism and interactions have we figured out. So we've done a lot of basic things on understanding how this works under normal conditions. But then we started to think about cancer, because it's well known that cancer cells need a lot of copper. But I know that there's no free copper. All copper is bound to proteins. So then the question becomes, what copper-binding proteins are important in cancer cells? And nobody had really looked into that. So we started to look at our proteins if they played a role in cancer. And then we found that this protein ATOX normally transports copper in the cell. We also found it at the edge of the cell, at the front of the cancer cells, suggesting that it played a role in cell migration, how the cells were moving around. And we found then that, you see some cells moving there? We can look at the cells under the microscope and track their movements, but we found that if we engineer away the protein from these cancer cells, the cells cannot move anymore. So this protein was important for cell migration. And why does that matter? Well, cell migration, movement of cells, is kind of the first step in metastasis. And metastasis is really what kills cancer patients. So that's what one wants to stop. So then we looked in cancer patients because we wanted to link this all the way up to real breast cancer patients. And here you have a plot of patient survival for breast cancer patients versus if they have a little or a lot of this ATOX protein expressed in their tumor cells. And you can see here in blue are patients with a little bit of ATOX only, whereas in red you have patients with a lot of ATOX in their tumors. And you see that there's decreased survival of patients when you have a lot of this protein, suggesting that the molecular reactions that we identified in test tubes and in cell culture actually is at play all the way up to patients. So if we can now, this protein cannot move a cell on its own. It's involved in some pathway here. So if we can reveal the involved players that mediate these reactions, maybe we can find new targets for attacking cancer metastasis. And I think this can maybe be part of a general strategy, because it seems like all cancer cells need a lot of copper in order to be to survive or to thrive. So maybe it can be part of a general bottleneck that we can tackle for all cancers. Another example I want to tell you about is about amyloid formation, and here for the protein involved in Parkinson's disease. So Parkinson's disease is the second most common neurodegenerative disorder. Alzheimer's is most common, and there's a lot of general features or similar features, common features between these diseases. It's just different proteins that forms the amyloids. But a lot of patients have Alzheimer's and Parkinson's. In fact, every third second, a person is diagnosed with a neurodegenerative disorder. That is like now. I mean, you can think about it. It's a lot of patients. And as I said, they're just increasing and we have no cures. So we looked at this amyloid formation pathway from different angles and tried to understand how other things modulate it and how we can modulate it to stop it. And one idea is that maybe Parkinson's, maybe other neurodegenerative disorders also, actually start in the gut. We have a lot of bacteria in the gut, and these bacteria help our bodies kind of, survive in many different ways and help us with a lot of metabolites in our cells. So we need this symbiosis with bacteria. But these bacteria that we have in the gut, they can also make proteins, and they have proteins that make amyloids also. So there are bacterial amyloids and there are human amyloids. But their idea is that maybe these diseases start from the gut and then they can transfer to the brain, and then in the brain the cells are so sensitive so they die. So we looked a little bit, we started to look at the proteins that form amyloids in bacteria, and if they can cross-react with the human proteins and what that means. And some of them can inhibit, some of them can trigger, so it will depend a lot on what the composition of your gut bacteria are. But then we started to think about food. Because food contains proteins, I said that before, but there's also some proteins in food form amyloids. And they are not degraded to the building blocks in the gut, but they remain as amyloids. And they therefore have an ability to escape into the human body. So parvalbumin is an abundant fish protein that forms amyloids in the gut, and that probably is the reason it survives the harsh conditions there and escape out into the blood. We know this because if you are allergic to fish, maybe somebody in here is allergic to fish. If you are, this is the protein that you make antibodies to. So it's an allergen. We knew it could come into the human body, so then we studied, can it interact with the human amyloid proteins? And we found that these parvalbumin amyloids can scavenge the Parkinson's protein and block it from forming amyloids. And this also worked on the Alzheimer peptide. So here is an example of how we can see that in an experiment. You see to the left, you see the Parkinson's amyloids. They are thick, right? You see some thick things there. In the middle, you have the parvalbumin, the fish protein amyloids. They are much thinner. And if I mix the two proteins, I only see the thin amyloid fibers, meaning that the human amyloid protein, Parkinson's protein, is not forming amyloids. I know you will ask me this later, so I put it up here, where you can, what fish has a lot of this protein in it. You can read that there, but a lot of common fishes. We only tested this in test tubes. So I don't know if this works in human, but maybe this can be a concept that can be used to use kind of formed amyloids as kind of scaffolds or scavengers to prevent human proteins from forming amyloids. Maybe we can make a pill of it with a lot of this fish protein, right? And that would be beneficial. This might also explain why fish is healthy. I mean, we always say that fish makes you smart, fish is good for the brain and so on. Maybe this is part of why that is actually true, that we block some kind of dementia reactions by eating fish. So with that, I want to come to my final research page here. What questions are we asking ourselves right now? What is the things that goes on in the lab right now? Well, I'll tell you two things. One thing is that these amyloids, they are a part of disease, right? And I told you they're toxic, they can spread from cell to cell in the body. They are large physical things that kind of can block the cells and damage the cells. But what we recently found is they can actually also act as enzymes. So they're not inert, they can do chemistry. They can catalyze chemical reactions. They are not really good. They are not as the evolved enzymes that we have in our bodies that do all the important functions. But an amyloid is going to sit in the brain for 20 years, so maybe that sloppy enzymatic activity can actually affect disease progression. And if that's true, that could really be important and a new way to target the diseases. Because metabolites, the energy metabolism in the cell is changing in these diseases, and maybe the amyloids actually contribute to that chemical change. Maybe even amyloids, peptide assemblies, could be thought of as primitive enzymes in the prebiotic world. Early on, when life was very primitive, maybe peptide assemblies could actually catalyze the first reactions. There's a lot of things that we can start to look into here that I think is really exciting. The other thing, if you think about cancer, is that we found another protein. It's called MIMO1. It's a cancer protein. People know that. But what we discovered is that it actually binds copper ions. And that seems to be a mechanism how this protein is an oncoprotein and helps cancer cells survive. Because, as I said, cancer cells has a lot of copper. So then there could be some free copper now and then. That's a bigger risk in a cancer cell than in a normal cell because you have so much copper. Then we have this MIMO protein that comes and scavenges copper that comes loose. So it don't have time to damage the cells, but instead MIMO protects the metal ion or protects the cell from the toxicity of the metal ion. So it kind of helps cancer cells survive. Maybe that can also be used as a new cancer target. In fact, there are more proteins. Today we know about 50 proteins in a human cell that binds copper, but more are being discovered as we speak. So I think this will just be bigger and bigger, and there will be more proteins that can bind these metal ions and do that for real function. So my overall motivation for all the research that I'm doing is really to try to find out new molecular knowledge about proteins and their actions and how that can eventually help combat human diseases. So my take-home message is, proteins are essential and fold to unique structures in order to function. That's the life part of this. But proteins can sometimes misfold and aggregate, and that can cause diseases. And amyloid dementia diseases are increasing in the world. Then you have the death pathway, because this is really scary. I also told you that folded proteins can be good or bad depending on the context. The copper protein ATOX, we need it for normal copper transport, but in a cancer cell, it can promote or facilitate metastasis, and that is not good. And we need to take into account the complexity of the cell, the crowdedness, all the other partners. I mean, it's a big, big soup of many things that can bump into each other. And most importantly, research is fun. Discovering new things that nobody else has discovered before, that's really exciting. And very important, it's all about people. Research is about people and interactions, and nothing of that I told you would have happened if I didn't have, my colleagues, my students. So therefore, I want to show you all my students. These are my graduate students and postdoc students over the years. I've also, in addition to these people, had many master students, undergraduate students, and also high school students that come and do projects in my lab. And this is my current research group, the ones that do the experiments right now at home. And of course, we need to thank funding because they pay the bills so we can do it. Genie is a project that I was running or started at Chalmers that I want to bring up because it took a lot of my time, and that's maybe why my research got slower for a while, but it was about gender equality. And I think that's really important to care about the academic environment as well as doing great research. So with that, I want to say thank you very much.
Thank you so much.